Hello everyone, my name is Allison. Today I'll be introducing you to your Bio20 project, where you'll be creating mutants in Arabidopsis using CRISPR-Cas9 as a way to investigate division plane orientation in plants. So I first like to start off by showing some examples of division plane orientation mutants. In this first panel over here is a TBO stain of wild type or normal maize epidermis. And what you'll notice is that the cells have these very regular shapes, and overall the tissue is pretty well organized. Here is an example of a maize division plane orientation mutant, which is mutant in the tangled 1 gene. As a result of these misplaced divisions, you get these unusual cell shapes, and you can see that the overall organization of the tissue is defective as well. I also have an example in Arabidopsis. This is an Arabidopsis root tip. Again, you can see that the cells have these really nice regular rectangular shapes, and that overall the tissue is well organized. Then this division plane mutant, which is a POC1, POC2 double mutant, you can see the division planes are misplaced, and you get these unusual cell shapes, and overall the organization of the root tip is affected. This has important implications for the overall growth of the plant as well. Here you can see a wild-type maize plant, which is nice and tall and healthy, and then the tangled 1 mutant has a growth defect. Now it has this short stature, it doesn't grow as well. You see the same thing in the Arabidopsis example. Here we have a pair of wild-type Arabidopsis plants, which have these nice long roots. And then if you look at these pairs of POC1, POC2 double mutants, you can see they have this short root phenotype as a result of defects in growth. Now, division plane orientation isn't only important for plants, it's important for animals too. For example, all multicellular organisms, like the C. elegans embryo, begin as a single cell, and they have to undergo a very ordered series of divisions to eventually give rise to a correctly patterned adult. If you're interested in medicine, division plane orientation is also very important in terms of cancer and metastasis. One of the genes I'll be talking about in depth, tangled 1, also known as TAN1, also shares amino acid similarities to this tumor suppressor APC. And when you think in terms of tissue, for instance, if you want to grow your epidermis, it's important to position your division to divide within the plane of the tissue. And then in other cases, you may want to divide outwards. Perhaps if you're a stem cell niche and you want to differentiate into a different cell type, the positioning of the division plane can be important for that as well. You can imagine if that gets misregulated and you have cells dividing out of the regular plane of the tissue, this could lead to the formation of tumors. And if you take that even a step further and you divide out of the tumor, improper division plane orientation can contribute to metastasis, this process where cancerous cells can leave the tumor and spread to other parts of the body. I'd like to talk a little bit more about division plane orientation in plants, because they have two additional structures that are specific to land plants. First is the pre-prophase band, also called the PPB, and the second is the phragmoplast. So the pre-prophase band forms prior to mitosis, and it's a ring of microtubules and associated proteins that forms at the edge of the cell around the premitotic nucleus. The pre-prophase band is actually disassembled upon entry into mitosis, and then these proteins that associate with the PBB remain at the division site, including tangled 1, and they serve as a molecular memory. So during telophase, when the phragmoplast forms, and the phragmoplast is an antiparallel array of microtubules that directs synthesis of the new cell wall starting in the middle of the cell and spreading outward to the site originally marked by the pre-prophase band. So the pre-prophase band, which is formed before mitosis even begins, actually serves as a marker for where the phragmoplast should construct the new cell wall. Now that we understand that a little bit, we can look at why tangled mutants have division plane defects, and it happens to be because of a phragmoplast guidance defect. So first I'm showing an example here of a time lapse of a wild-type maize epidermal cell undergoing division. Now this is a cross-section, so what you see here, even though the pre-prophase band is actually a ring, it looks like two bright puncta. What you'll notice is that it disassembles, the spindle forms, and the new cell wall is constructed at the site originally marked by the pre-prophase band. indicated by those blue arrows. However, in a proportion of divisions in the Tangled 1 mutant, there are fragmoplast guidance defect. So here these blue arrows indicate where the pre-prophase band is, and you can see those bright puncta. But what you'll notice is that when the new cell wall is being built, it veers off. So the fragmoplast is misguided away from where the pre-prophase band was, and this causes these misplaced divisions. I've been talking a lot about maize, but what we're actually going to be working with is this plant called Arabidopsis. That's because Arabidopsis is a really great model system. Unlike maize, they are very small plants, which makes them much easier to grow and maintain. They also have a short generation time. You can think of this as their lifespan, and that's useful because you're able to collect seeds more quickly, and you're able to study multiple generations of plants much more quickly. They're also very well studied. They're very commonly used in plant biology labs, so they have a very well sequenced genome, and they also have a lot of great molecular tools available that are not available in maize. So it's actually pretty easy to generate Arabidopsis mutants and transgenic Arabidopsis. I'm talking about Arabidopsis, but for a while we actually weren't able to study Tangled in Arabidopsis because the Tangled 1 single mutant in Arabidopsis has no discernible phenotype. So here we have an example of the wild type seedlings, and then we have the Tangled seedlings. These pair over here. You can see they grow the same, and I can tell you the adult plants look the same as well. When you look at these propydium iodine stained root tips, which let you see the cell walls in these root tips, you can see the wild type is nice and well ordered, has these very nice rectangular cells, and you see that for the Tangled 1 single mutant in Arabidopsis as well. It has no defects in division plane orientation. Now, kind of recently, with the help of a collaborator, we identified a TAN1-AR9 double mutant, which has a division plane orientation defect. So it has a synthetic mutant phenotype. So what that means is that we have our TAN1 single mutant, and what you see, it looks normal, it looks like the wild type. AR9 is another division site localized protein, and hopefully what you can tell from here is that they too have no division plane orientation defects. However, when you have mutations in both of these genes at the same time, now you see something that resembles that Tangled 1 mutant in maize, where now you have these unusual cell shapes as a result of misplaced division. When you look at the plants as a whole, here you can see the wild type and single mutants, they all grow just fine, but when you have mutations in both genes, TAN1 and AR9, now you see that stunted growth phenotype. So I keep saying synthetic mutant. What does that actually mean? Well, this is what happens when you have a mutation in 2 or more genes and it creates a mutant phenotype that is more severe than the single mutant phenotypes combined. So if we knew nothing about the TAN1-AR9 double mutant and I crossed the TAN1 mutant to the AR9 mutant, I might expect, since these guys look wild type, I would expect maybe the double mutant would look the same. I would have no reason to believe it wouldn't also produce a normal looking plant. But that isn't the case. You can see that the TAN1-AR9 double mutant, so when you combine these two normal looking plants that have mutations in these genes, you get something that's a lot worse than just the single mutants. So what causes this synthetic mutant phenotype? Well, one way to think of it is that you can think of TAN1 and AR9 as 2 arms or pathways that can do the same thing. So as kind of a metaphor here, I have a knight and he fights using a sword, and he has 2 arms, his TAN1 arm and his AR9 arm. And you can think of this knight here as a wild type plant. He has both pathways working just fine. Now if you get rid of one gene, or one arm, it's still fine. Now this knight is like an AR9 single mutant. You can see it's missing the AR9 pathway, but he can still hold his sword thanks to his TAN1 pathway. So this is just like how the AR9 single mutant can still correctly place its new cell walls because it still has TAN1, and this knight can still hold his sword. However, if you get rid of both, so if you get rid of both arms or both genes, Now this knight is like the tan1 air9 double mutant. He can no longer hold his sword, and he can no longer fight. Just like the tan1 air9 double mutant no longer has either gene, so it can no longer correctly place its new cell walls. Just to re-emphasize that, you can think of the one-armed knight as a tan1 or air9 single mutant, where if you lose one, you still have one to compensate, and as a result, your single mutants look just like the wild-type plant. And now you can think of the no-armed knight as a tan1 air9 double mutant. So now he has neither pathway, just like our double mutant doesn't have tan1 or air9, so now you get this growth defect. Now biology is rarely ever that simple, so we're going to take this metaphor a step further, where now we're going to think of each arm as a different pathway, where this arm is the tan1 pathway, and this other arm is the air9 pathway, and each pathway has a different set of genes involved. So here we have our arm, and maybe tan is this bone, and then all these other bones are a bunch of different genes. And we have a long list of potential genes that we want to study that we think may be involved in the tan1 pathway. But now we have this question, how can we actually figure out from this long list of genes which are actually involved in this pathway and which are actually important for division plane orientation? Well, the way we're going to do this is we're going to break some of these bones by creating mutants. And we're going to do that using this CRISPR-Cas9 technology to mutate each of these genes in our list. So when I say mutate, I mean basically get rid of these genes, just like how here we're breaking each of these bones. And then we're going to see whether or not it's needed to correctly position division planes in plants. Another caveat to this is that we specifically want to mutate the air9 single mutant plants. Why? Because we saw earlier that these pathways can compensate for each other, and we just want to focus on the tan1 pathway. So if the air9 pathway is still there, this arm is still here, we won't be able to focus on just one thing, because this guy will just compensate. So we're studying the air9 single mutants, or the single arm knight. So the way this will work, if we're mutating these genes and getting rid of them, if that gene turns out to be important for division plane orientation, here's what we'd expect to see. So we have our air9 single mutant, represented as this one-armed knight. Let's say we mutate a gene, so we're breaking this bone in his arm. If that gene was important in the tan1 pathway for division plane orientation, we'll see a double mutant phenotype, where his arm is too busted to hold his sword anymore, so he might as well just be a knight with no arms. Or in the case of our plants, that gene was important for function in division plane orientation, so we see a double mutant phenotype where we see this growth defect. Now, not all these genes are well studied, and we don't know what the outcome will be, so it's possible that when we knock out some of these genes, we'll see they are not important for division plane orientation. So, again, we have our air9 single mutant, our one-armed knight. Let's say we break this bone, and it's like breaking a pinky finger. It's like whatever, you can still hold on to the sword. So if that gene is not in fact important in the tan1 pathway for division plane orientation, we'd see no change in the phenotype. So he'll still be able to hold his sword, and the plants will just look like a regular happy air9 single mutant. So with that, I just want to reiterate the goal of the project. So we want to identify genes involved in division plane orientation in the tan1 pathway. The way we're going to do this is by generating mutants from our long list of genes. We're doing this within the air9 Arabidopsis single mutant. And what you guys will be doing is that you'll each get a gene, and you'll help design guide RNAs for the CRISPR-Cas9 system to target these genes and knock out their function. And don't worry if you don't know a whole lot about CRISPR-Cas9, we'll have another lecture on it in the future when we get there. So the basic flow of the experiment, our experimental design, is that you guys will help us design a bunch of guide RNAs for all these different genes. We'll clone them into a vector that has the Cas9 coding sequence and selectable marker. And then maybe we'll talk about this next process. We'll actually use a process called floral dip to introduce our CRISPR-Cas9 system into the air9 single mutants. And then what we'll do is screen those CRISPR Cas9 mutants that have their genes knocked out in the AIR9 single mutant background, and we're interested in finding plants that have a severe growth defect compared to the AIR9 single mutant, because that will indicate that that gene we knocked out may be important in the TAN1 pathway for division plane orientation. And with that, I'd like to thank you all for your help. Thank you in advance. If you have any questions or comments about the project, feel free to email me at this address right here. And in the process, we'll be teaching you guys lots of other cool stuff in the lectures to come. We've already started on general primer design, and we'll also do some activities on phylogeny, and then finally we'll get to the guide RNA design, and then hopefully we'll come up with some more cool stuff to show you guys. And then over here is just an image of my wonderful lab mates, past and present. I'm the one in the pink hat here. You probably won't see me around nowadays since we're all cooped up at home, but that's what I look like. And then hopefully we'll have an appearance by my PI at some point in the future. Her name is Dr. Carolyn Rasmussen. You can see her right there in the back. And that's it for now. Thank you for your help, and I'll talk to you guys some more later.